I'm Jade Mitchell. I'm a professor at Michigan State University in Biosystems and Agricultural Engineering. I'm also the Associate Department Chair. What I do is a big word, quantitative microbial risk assessment, but mainly I'm looking at pathogens that impact water quality and food safety and how they move through the environment and how predicting those risks can help us develop better systems that are more resilient. We had really responsive partners. We did a workshop with them so that they understood what we wanted to do with the data that we were collecting in their systems, and also to find out what their concerns were so that we were a resource to them and they weren't just a source to us. The framework has always been to look at a single stressor at a time, but they actually exist with many other stressors. So understanding both chemical and microbial safety is important because we use chemicals to control microbes, right? So if we're going to do disinfection, we want to understand, is there a chemical risk from that disinfectant or those disinfectant byproducts that is causing more harm or a different type of harm than the microbe itself. I'm going to talk about risk assessment and give you a bit of an introduction, and then I have 3 case studies from some current projects that have been completed in my lab. And you can see based on the title, I think risk assessment is a tool that we can use to advance resilient water systems. So I'm going to try to highlight where what we've learned from the case studies support that. My work is in quantitative microbial risk assessment, which in itself consists of several disciplines that are put together to establish a computational framework to understand how pathogens move through environmental media and eventually into contact with people, and then how those contacts interact with their body and produce human health risk. It follows a 5-step paradigm that I have there at the bottom of the slide: hazard ID, dose response, exposure assessment, risk characterization, and then finally risk management. This isn't really a new framework for QMRA, but QMRA really just established itself in 1999, which for a science is fairly new. That framework, or at least the 4 steps, hazard ID, exposure assessment, health effects or dose response, and risk characterization, come from the National Academy's 1983 framework document for chemical risk assessments. But as you can imagine, microbes pose different challenges in the environment and in water especially that chemicals don't. So in our first step, hazard ID, we want to identify the type of microorganisms or disease endpoints associated with those microorganisms. Sometimes we know them. A lot of times we have to pick a target, and other times we have to do retrospective analysis. Exposure assessment relies a lot on monitoring, but as I like to tell my students, you can't measure everything everywhere all the time. So a lot of the work here is through integrated models that evaluate fate and transport, especially through water systems, of how those pathogens are moving and eventually coming to the tap, or in recreational exposures, the place where, or the point of where people are coming in contact with it. Health effects assessment or dose response. This is where it's really the yardstick of risk assessment. It's where we develop a mathematical relationship between an exposure dose, the amount that you're exposed to, and the probability of some adverse health outcome, like infection or illness or mortality in this case. And it relies on a lot of animal studies that we extrapolate to humans, just like in chemical risk assessment, but occasionally for those pathogens that are not too dangerous to work with, we have human feeding studies that inform these. And then finally, risk characterization. Generally, we use some type of simulation, like a Monte Carlo simulation approach, to be able to estimate the risk, but also the uncertainty and the variability. So as you can imagine from what I've described as the inputs, the exposure assessment and the dose response, there is a lot of variability and uncertainty depending on different environmental pathways, conditions, and host susceptibility or effects. Risk assessment itself fits into this bigger framework of risk analysis, which also includes risk management and risk communication. And I would argue that for microbial risk, we have more of an integrated approach than we do for chemical risk. Because in general, for chemical risk, we're looking at long-term, small exposures that result in health outcomes, at the end of your lifetime. But microbial risk pose acute outcomes, like when you get gastroenteritis, sorry for saying that while you're eating, but right away, right? So some communication or management is required because outbreaks happen and people know about it. All right, so here are 6 different areas where I think advancing microbial risk assessment can help with water security. Down here at the bottom, the first thing is through system mapping. So in order to determine the quantity of the pathogen that someone's exposed to, we have to understand the whole system from source to tap if we're talking about drinking water. And a large part of my talk will focus on drinking water. So it considers source water protection, sources of contamination, treatment, what's removed, what's not removed, and then release into distribution systems, which I'm going to touch on a lot. This will allow us to identify system vulnerabilities just because of that mapping and that evaluation of points of contamination. Then we do the actual risk assessment, which I just described. That supports sustainable innovation. So if we want to put in a new technology or a new material, we can evaluate what the risks are across those exposure pathways. That work then leads into usually some risk comparison. If there's a bright line approach, like we can say, is the risk above what's acceptable, like 1 in 10,000 infections per year for drinking water? If not, how do we compare it to other risks that are acceptable? Or is there another approach we can use to determine if risk is acceptable and then do those comparisons to support decision making? Some decisions are related to treatment, so we can evaluate treatment performance based on how they reduce risk within the system. We can also prepare for emergencies. QMRA is used for water safety planning and is one of the approaches that WHO, the World Health Organization, emphasizes for water safety planning in other places besides the US. And then finally, I'm going to talk about integrated assessment, which really hasn't been fully utilized, but is an approach that we're starting to work on now, where we can integrate both risk associated with microbes and with chemicals. And you can think of antimicrobial resistance as maybe one of those challenging areas where we have both chemicals and microbes, and we have to balance those risks. So what are the issues that we need to address? These two are not very new. Aging infrastructure. So repairs introduce lots of new materials within a system that is established and has been for a while, but this is needed to, we can't repair the whole system all at once, so maybe it's obvious why that's needed. One of the other things is source water changes, like there are climate effects that require systems to choose a different source water. And we know from Flint, Michigan, I'm from Michigan, so that one's usually associated with the type of work that I do, that changing source water can certainly impact the scale that is already within your system and cause leaching of contaminants into it. emerging and re-emerging hazards. So I don't know, I struggle with using the word emerging with Legionella because it was always there, but there are some things that are happening within systems, or maybe we're just able to capture those illnesses more that point to needing to be able to manage pathogens that we haven't previously managed. So our drinking water standards currently are focused on gastroenteritis causing agents. But many of these opportunistic pathogens, like Legionella, are inhaled. And that's a different exposure route that we need to evaluate before we could come up with standards. Disinfection itself is the way that we would prevent microbial levels, but disinfection needs to move from an art to more of a science, because we found that once we've changed disinfection protocols to control for opportunistic pathogens, for example, so lots of systems have moved from using chlorine to using chloramarine, that the disinfection byproducts that are developed are actually more toxic than those that are regulated and were caused by chlorine. So there's a lot of work there. And then the final thing would be social structure. So I mentioned that maybe we're just capturing more data about Legionella or other types of infections, but also our population's changing, and in some ways they may be more vulnerable to microbial infections. So if we just look at public health cost, and these are the latest data that I could find that were published in 2021, the majority of the cost associated with waterborne microbial infections are from these opportunistic pathogens. So NTM is non-tuberculosis mycobacterium. Legionnaire's disease is caused by Legionella. Pseudomonas is caused by Pseudomonas aeruginosus. And these rise to be much higher than Giardia and Cryptosporidium, which are regulated, or that we use. They're not regulated, there's no limit, but they're used to establish log removal in drinking water treatment. So a lot of the work that I've done in the past 5 or so years is focused on these opportunistic pathogens and understanding their risk and how we can determine vulnerabilities and then find ways to control them. So what is an opportunistic pathogen? Like, I named the pathogens, but they're pathogens that pose harm because there's an opportunity to do so. And those opportunities usually present as more susceptible populations. So in the general population, the risk of Legionnaire's disease is much lower than it is in elderly populations or for people with comorbidities. And these pathogens are actually in our system already. They're fairly ubiquitous in even well-maintained distribution systems. But they are associated with certain environmental conditions, like low chlorine residual, or disinfectant residual if it's not chlorine. They're associated with warmer water. They proliferate inside of amoeba, so understanding the microbial community of the water, not just looking for individual species, is important. They grow in biofilm, so they're also generally found in certain fixtures, like in the premise plumbing. And premise plumbing is not regulated. The distribution system, or the point of entry to the distribution system is regulated. So we need to be able to understand what happens after that so that we can control these risks where we do have some control over water quality. So I'm going to talk about or the 3 projects that I've picked as case studies, but all 3 of them, consider both chemical and microbial risk together. And comparing these lets us sometimes, just look at what the actual risks are because we might think it's one thing and control for one thing, but cause an unintentional consequence. They also allow us to optimize our system because we want to reduce both. And then finally, in terms of treatment, they can help us choose in decision making what appropriate or needed treatment technologies are. So this is the first case study. It's a project that was completed in 2022. It was done in collaboration with the universities below, and I listed my co-PIs, starting with Andy Walton at Purdue, who was the PI of the whole project, and I was the PI at MSU. I feel kind of lucky I get to do computational and modeling work, but I don't have to go out and do the water chemistry or the microbiology, so I always acknowledge my collaborators first. The goal of this project, or the three objectives I'll focus on, are improving the public's health understanding of decreased flows. So water conservation is a good thing in general, right? I think you probably all agree water is a precious resource. But the water conservation policies were really directed by energy policies and didn't really look at the impacts of what decreasing our use of water would do to water quality. So we end up with water that's stagnated or just sitting in pipes, which isn't necessarily a good situation because we don't understand water stability that well, like what's the shelf life of water? The second thing that we wanted to look at is, could we look at factors and interactions that affect drinking water quality, like system characteristics or source water or the piping system design, these external factors that might be risk factors for predicting when there might be a water quality problem, so that we could inform designers and reduce these risks. And then the third thing was to develop a risk-based decision tool that could be used by people other than modelers and scientists and researchers. So this project, or at least the data that I'll show you, mainly focused on the Renew House, which is a Whirlpool net zero home that's on the campus of Purdue. And it's one of the most monitored homes in America because it has so many sensors. There are flow meters and thermocouples, and it has 1 second real-time readings for many different metrics that you would want to know in a home to evaluate water usage, water temperature, where are those gradients occurring that create like I would say the Legionella friendly zone within the home, and then certainly how much energy is being used. So because I'm a modeler, I like this slide, but every time I look at it, I'm like, oh no, is this what we really proposed? This is sort of the framework for the problem, or for the project, right? We started out looking at water demand, and not just in the peer-reviewed literature, but in the gray literature. Then we went out and we took field measurements, mainly in the Renew House, but also in some commercial office buildings. We measured many different water quality parameters, as well as opportunistic pathogens like Legionella and Pseudomonas. And then we wanted to develop a predictive model that would be able to develop many simulations so that one could look at, if you change things like pipe length or temperature or water usage, what would you expect the water quality and the opportunistic pathogen levels to be? And that's kind of what I'm showing here in the big, busy slide. We used risk assessment finally to determine, even though we have these measurements and all of this data, what does it really mean for human health, and do we need to do something about it? And that's part of the decision tool. So there were lots of presentations and publications from this. If you go to that plumbingsafety.org website, that will take you to a website where everything is listed, because I won't have time to go into detail for everything. But this is one of the ways that we were able to provide the information to the public and to decision makers and designers through this repository. We were mainly focused on Legionella after finding that it was the most common opportunistic pathogens of those that we had measured. So we started off developing novel models for other opportunistic pathogens, but after several years of monitoring, we realized that Legionella was the thing that we needed to focus on. So we looked at how the plumbing design and other water quality parameters could help inform our estimation of Legionella. The paper that I've referenced there at the bottom is one of my postdocs. We used a few different statistical approaches, like a GLM. We looked at a Bayesian regression method as well. We used principal component analysis to determine which factors were most contributing to Legionella. And overall, like through all the methods, we found that it was water age. So water age isn't really something that's easily measured. It's predicted, right? But I mean, it's the hydraulic retention time. But since this is a system that has constant inputs and outputs, every amount of water doesn't have the same age, and we don't always know how much mixing is occurring. We did end up developing our own water age model and comparing it to EPA net. And we had okay agreement. That's another paper that I didn't reference here. But we did use EPA net and the MXX file to develop that hydraulic water quality model, where one would be able to put in different types of pipes, different demands. I'm looking at temperature, water quality, and there were actually 256 scenarios, so trying to think of all of them on the top of my head is probably not a good idea. I should refer you to the paper for that. But the idea here and the end result is that you would be able to change parameters and develop different scenarios and look at how that would affect your ending water quality in a predictive approach to inform the decisions that you would want to make about pipe design. There are 8 of these models that were developed. There is a paper that quantifies the results of those 8 models, but they were ultimately put into 2 different modules of a decision support tool. So the first one predicts water quality based on all of those parameters that I just mentioned, and then the second one takes the Legionella concentrations that you would expect from those simulations and will calculate risk for you. So we can assume that for Legionella, that 1 in 10,000 infections per year is acceptable, because that's the guideline that we use for drinking water quality. But in actuality, we know that that's based on gastroenteritis and not inhalation. So something to consider if you're going to use the tool. All right, the second case study was another EPA-funded project. Again, these are all my collaborators who are kind enough to take my crazy experimental designs and go out and take samples and analyze them for microbes and chemicals so that I can have great data. This project focused on distribution systems rather than premise plumbing systems. And here, again, we were focused on opportunistic pathogens, but we wanted to know for regulators and for water utilities if we could identify system characteristics that would predict occurrence and co-occurrence of disinfection byproducts and opportunistic pathogens together. So here's the goal that I just mentioned. I should say that unfortunately this project was terminated early with the EPA grant terminations, but we do have a full years of data and the pilot, so analyzing and putting this data together is part of what we're working on now. We partnered with 25 different utilities across the US, representing most of the regions, 9 of them, but not all of them. We sampled at 6 different locations within the distribution system. We took, I think, 2 or 3 samples from each, and as we progressed into the following years that we're not going to be able to do, we were planning to take more samples at those facilities where we found more occurrence and co-occurrence. But we took plant influent, we looked at the point of entry into the distribution system, and then we let the utilities self-identify 4 additional sites to sample based on where they thought their mean and max water age were and where they thought they had low chlorine residuals. These samples were analyzed at Clemson for the DBPs, and we looked at 11 regulated DBPs and 41 unregulated. I mentioned earlier that the unregulated DBPs, at least some of them, may be more toxic than those that are regulated. So we wanted to have a good understanding of the mix of DBPs that were there. Our samples for microbial analysis were sent to Tulane, and we looked at Legionella using a number of different approaches, and as well as Pseudomonas. So here's the results there. For aggregate analysis of DBPs, we found that we could take a total of all of them, and that THMs, HAAs, and HALs were the most common. I don't know if that's good news or bad news. Probably when you're in the risk world, there's no good or bad news. It's just the data. What we found is that in the majority of these cases, the levels were not exceeding the regulated limits for these DBPs, and these are regulated, so we had something to compare to. But we also realized that just looking at the total aggregate occurrence doesn't really tell us if we need to do something about it. So one of the metrics that can be used to look at toxicity is cytotoxicity. So here I'm showing what we found across all of those plants based on cytotoxicity, and what rises to the top are HALs, HAMs, and HANs, not total halomethanes, which are already regulated. And there really were only a few plants where we had much higher numbers. The other thing that we wanted to understand over time is where are we finding these things within the drinking water system? Those yellow dots are areas where there's low chlorine residual, and the max residence time are the green dots. Most of those are buried, but you can see some of them. And then the middle range are those blue dots. So definitely the places where there's low chlorine residual, that may or may not correspond to the place where the utility thinks that they have the maximum residence time or water age. For the opportunistic pathogens, there were 577 samples found. The majority of cases where we found occurrence for Legionella and for Pseudomonas, I'm sorry, not for Pseudomonas, for Legionella were in the distribution system and not in the source water. For Pseudomonas, it was in the source water and not in the distribution system. This was actually consistent with the findings from that previous EPA project, though we were looking at distribution systems this time. One of the things that we wanted to look at here were based on the assay that we used to measure Legionella, did we find differences? LegAlert is one that is easy for utilities to use on their own. They don't need a full-scale microbial lab. but the culture-based method does need to be performed in a lab. Unfortunately, we didn't find good agreement here, but the literature doesn't have consensus on this. So the important thing here is what tools you use to produce the data or the quantified microbial data that goes into a risk assessment. It does matter how consistent it is, and that adds uncertainty to risk assessments. We did find a good agreement between the Legionella species culture method and Legionella pneumophila. So there are many Legionella species, but Legionella pneumophila is the one that causes Legionnaire's disease primarily, and it's the one that we're most concerned about. I won't go through the whole data dictionary, but basically we wanted to identify system characteristics that would help us predict co-occurrence and occurrence of the hazards that we were concerned about. So we identified those for each of our different plants and locations. And then we had this conundrum because there's no metric for co-occurrence. They're both there, that's a binary variable, but both there in a quantity that we care about is not something that had previously been defined. So that was our dependent variable, and we had to develop that through the study. And here, for co-occurrence, we considered Legionella species that were above 1 CFU per 100 milliliters, and cytotoxicity because it's representative of all of the disinfection byproducts that were present. That's greater than or equal to 1. So things that popped up that we knew about were that low residual would be one of the determinants, that warmer temperatures, so those we found in September, at the end of summer, where the waters had a chance to heat up. And this was actually pretty close, whether the system used chlorines or chloramarine. It was higher for chlorinated systems, but you should note that we didn't have many systems that were using monochloramine. We put that all into a generalized linear model, including that metric that we had to invent as our dependent variable. And we removed our correlated variables like population and flow rate, which were expected to be correlated, so that we would just use one variable representing size of the plant. What we found were actually the same things that we would expect, that pH and nitrates, bromides, iodides, and the size of the system were the biggest determinants of Legionella that would be present in the plants or in the distribution system in the locations that we measured. So while you do a study and you want to find something new, actually it's great if you can validate at full scale in distribution systems across the US that the same thing that would be predicted at bench scale is what happened. So here I basically presented a risk factor approach because we didn't necessarily calculate the exact risk, but we determined that there are system characteristics that we can use that would be predictive of what the risk for occurrence or co-occurrence would be. We do want to do a full risk assessment, and that's what we plan to do with this data in the next year or so. It's going to be harder because the project was terminated, but we have all this great data, so we're going to use it anyway. And we're going to go through that process that I described, the 4 steps for characterizing risk. But risk in itself is not a metric that you can directly compare, right? It's a probability of an illness, but not all illnesses are equivalent. They all have different severities and durations. So we need to translate that risk to a disease burden in order to do a comparison. And this kind of sets us up for comparing microbial and chemical risk also. I mentioned already that one is acute and one is chronic, but understanding disease burden is a metric that could put them on the same scale. So we know what our hazards are in this case. I won't go into detail here. We need that yardstick of risk, those dose response models that I mentioned, and we have them for the pathogens and for the regulated disinfection byproducts, but part of what we're doing now is developing them for the unregulated. Excuse me. And this is work that depends on evaluating many, many different animal studies and toxicity endpoints to understand what that risk would look like. So we've done a systematic literature review. We have lots of data that we can use to develop these. This is a next step that I won't get in the weeds on for the sake of time, because I want to leave time for questions. In this case, we did the monitoring, so we have the occurrence and we have the concentration data at the points that we're interested in, so we don't need to do much exposure modeling here, but there might be some environmental conditions that we would want to consider before determining whether our risk assessment is predictive. And then finally, we're going to do the risk characterization and consider the uncertainty and variability in our inputs so that we have a distribution for our outputs before we translate that into disease burden. And I should mention there's only one study that has done that translation, looking at microbial and risk and for disinfection byproducts. This was actually out of my graduate advisor's lab, so not surprising. We still do very similar work. But in this case, they looked at the comparison on a yearly basis. So for a given year, you don't have to consider those two different temporal scales that come into play that I've already mentioned, right? Over a lifetime, if you're drinking the same water, you're going to have multiple outcomes or acute illnesses associated with the risk. But for the chemical, you're still looking at the single lifetime case. So I'm going to move on to the third study where we address this and we were able to do this comparison. In this study, we're going to switch focus from drinking water to an agricultural reuse case. I like to tell my students that food is water because a lot of the work is water, but it ends up being used for food. So here's one of them. Again, I want to acknowledge my collaborators. This work wasn't done in the US. It was through collaboration with Politecnico di Milano in Italy. And I'll talk about some of the differences there, just based on regulatory regions. So we wanted to compare indirect versus direct reuse for wastewater. And we know that indirect is de facto. It's what we do in the United States. We treat our water, we discharge it to a surface water body, and then it can be extracted later downstream for multiple uses, including irrigation. There's many positive effects to direct reuse, and especially in this case, because in the EU, the water's not just treated, but it's highly disinfected, sometimes using UV, and then it's discharged into a canal. So while we couldn't look at the whole suite of contaminants, we wanted to know, based on certain microbial pathogens and those that we would expect to cause the biggest food safety or water quality hazards, do we have a larger risk from the indirect case or the direct case? And again, for a certain suite of emerging contaminants, for the chemicals, which is worse? There aren't a lot of studies that look at this for food either. So yeah, the systematic literature review is there, and that's the DOI that I'm showing on this slide. And this, I think, is what I just described, but I will tell you what our hypothesis was. So in the direct case, the water would be treated and go directly through some pipe system for use for irrigation. We would expect, because of the disinfection, that there would be minimal microbial risk, but there might be some chemical risk from the residual emerging contaminants that were not treated. In the indirect case, we expected the opposite. So the water's treated and then you're discharging it into surface water body. Because of dilution, you may have lower chemical contamination, but because you're putting it in a surface water body, there's opportunities for new microbial contamination or growth. I'm running short on time. There was a sampling campaign upstream after disinfection and of the effluent. And I've highlighted here that these are the pathogens that were measured, but they were measured like just based on presence absence. What was quantified was E. coli, so we had to determine what the ratios between the indicator and the pathogens were, and this happens like in so many cases because we still use indicators for water quality. We don't commonly measure specific pathogens. And then we also measured 14 contaminants of emerging concern. This was the framework for the risk assessment. We considered lettuce because it's eaten raw, so that would give us the most conservative case, rather than something that's cooked and the microbes die, of course, and then the risk should be negligible. And we're going through the entire risk assessment process that I talked about, and then translating to disease burden. So we're going to use the DALI, the Disability Affected Life Year, which is the metric that puts things on the same scale for measurements because it considers disease severity, length of disease, and it can also consider life lost as a result of disease. For the pathogens, we generally look at those on an annual basis. I've already mentioned that we have like a nice bright line for annual risk of infections for drinking water, but we don't consider those over a lifetime, but though they're occurring over a lifetime. For the chemicals, we do consider those over a 70-year life period, and for those that have regulations, there again is a bright line. We have a reference dose that's developed through those dose response models that allows us to compare and know whether we're above or below that. So what we did is we calculated our risk for several reference pathogens and for the emerging contaminants. I focused here on 3 that are pharmaceuticals, and we looked at them over a year, and it looked like the chemicals were producing the greatest number of DALIs or were the greatest risk. Then we considered how we could look at this over an entire lifetime because the infectious agents are causing risk over the entire time period. And for the DALI, you can just multiply it. You can't multiply a probability, so it was a little bit easier in the math too. If we looked at the 2 types of risk over the 70-year period, how they would compare. And you can see from the scale of the slide that our microbial risk ended up being much higher than the chemical risk that we evaluated for the emerging contaminants. This is a single case for certain reference pathogens and for certain emerging contaminants, but what it does is it establishes a framework where we can look at both chemical and microbial risk in an integrated framework together. I think I said all of this stuff. So hopefully what I've mentioned or shown in these studies at least brought up in your mind and some thoughts about how risk assessment might support water security in the way that I've mentioned. I did break it down for you, but I won't read all of these slides. Can we share these? Okay, I'll share these slides, which put the results and conclusions from those studies in the context of the 6 pillars that will advance water security using risk assessment. Thank you.